and your location, and we will love to know where everyone is joining us from. And please also keep your microphone muted during the presentation and kindly use the chat Q&A section for your question. Once again, you're welcome to this webinar. And now I would like to it is my honor to invite to the webinar chairman, who is the president of the Microtoxicology Society of Nigeria, and is our very dynamic and distinguished professor, Professor Charles Adetunji, to deliver the welcome remark. Sir, we are honored to have you with us. Professor Charles Adetunji, our president.
Okay, good evening.
Thank you.
Good evening.
Good evening,
my
sir. We can hear you.
Professor, I'm so delighted and excited talking to you today. Let me get started. It has been a couple of days, months, we've been on a series of webinars, and it's highly exciting having distinguished scholars, professors, and industrialists, and so many eminent people on board. Today's webinar is another horizon that's going to change our perspective, how the issue of aflatoxin, mycotoxin is going to be resolved. So I'm so sure all of us is going to go home loaded, refreshed, with a dynamic mind to change the level of food insecurity in Nigeria. If you are going to get it, we open our eyes. Without mixing words, I want to appreciate the ex-official, Professor Yamisi, that just talked, all our board of trustees, all the ESCOs, and the directorates in charge of webinar, and our distinguished scholar that just returned from China, from FUASI, Federal University of Agriculture, Irabiji. She's an HOD, elaborate scholar, distinguished scholar. I don't want to preempt a citation, just to be done by the moderator. So on this note, I can cite so many vice chancellors amidst us. I see some senior professors. I want to say welcome to this webinar. God bless you and see you at the top. Have a fulfilling and refreshing webinar. Thank you very much.
Thank you very much, sir, for those insightful and inspiring welcome remarks. We sincerely appreciate your leadership and your commitment to advancing the works of Microtoxicology Society of Nigeria. Ladies and gentlemen, we will now proceed to the main scientific presentation of today's webinar. It is my pleasure to introduce our distinguished speaker, Dr. Sholabomi Olaitan Ayoade, and I'm privileged to read a citation. So Dr. Sholabomi Olaitan Ayoade is a lecturer in the Department of Crop Science of the Federal University of Agriculture and Development Study, Iragbiji, Nigeria, with a distinguished career spanning advanced agricultural research, academic leadership, and international collaboration. Dr. Ayoade stands at the forefront of innovative crop protection strategies. She earns a PhD in plant pathology from the prestigious Xinjiang University, China, where she studied under a Chinese government scholarship. She also holds both a master's and a bachelor degree in crop protection from the Federal University of Agriculture, Abeokuta, graduating as the best graduating student of her department. Following her doctoral studies, she was awarded the highly competitive International Postdoctoral Exchange Fellowship through the Talent Introduction Program at the Institute of Biotechnology, Xinjiang University. Permit me if I don't pronounce it well, where she served as postdoctoral research fellow until early 2025. Dr. Ayoade's research specializes in nanobioprotectants, plant pathology interactions, and the green synthesis of nanoparticles for agricultural defense and safe food consumption. Her groundbreaking work has resulted in 2 authorized international patents for the biogenic synthesis of nanometal oxide and nano zinc oxide using bacteria, supernatant, and plant extract. An exceptional prolific researcher, She has authored numerous peer-reviewed papers in high-impact JCQ, QI journals, including the Chemical Engineering journals, Journals of Hazardous Materials, Ecotoxicology, and Environmental Safety, to mention but few, with an IEH index of 21, 110 index, 30, and total citation of over 2,000. Beyond her research, Dr. Ayade is actively engaged in shaping the next generation of scientists. She has instructed undergraduate and international graduate courses in both Nigeria and China, and currently serves as departmental coordinator at FUDATSI, contributing heavily to the global scientific community. She serves as an editorial member for Plants, a guest editor for the International Journal of Molecular Science, and a verified peer review for leading publishers such as Elsevier, Frontiers, Springers, Natures, and Wiley. Dr. Ayade's dedication to sustainable development goals and nanotechnology continues to drive impactful solutions for global food security. We are particularly pleased to have Dr. Ayoade with us today to share our insight on these important and timely subjects. And we know that the topic of today, as I've said it, is on fumonisin. Dr. Ayoade, on behalf of the Mycotoxicology Society of Nigeria, we welcome you warmly, and the floor is yours. Thank you.
Thank you, Prof. Good evening.
Good evening, Doctor. We are happy to have you.
Same here, ma'am. Thank you so much. I'm trying to share my screen. Okay. Okay. Can we all see my screen, please?
Yes, I can see it. I don't know if others are seeing it, but I can see it.
Okay. All right. Thank you so much. It's an honor to be here to present on this wonderful topic. I appreciate my Deputy Vice Chancellor, Professor Charles Adetunji, for this privileged opportunity you granted me. And I'm so much indebted to you, sir. Thank you very much, sir. I appreciate yourself for citing me and counting me worthy to come and give the presentation at this honorable international society. I'm glad for this platform, sir. And greetings to all my colleagues, my senior colleagues, my researchers all over the world. Good evening from here. Good evening from Nigeria. I hope we will have a very interesting discussion time. So now I will be presenting on fumonisins, a ticking bomb needing urgent intervention, its modern strategies, and for mycotoxin control. So my section outline will include, I'll be discussing on the background, introduction, the producers of fumonisins, the impact of fumonisins on man, plants, and the world, the economy on its own, then the intervention strategies that have been used all over this time, then the modern intervention strategy that is in place, then we will be concluding. So let's kick off. Fungal disease and mycotoxin, they pose a significant threat to human, animal, food safety, agricultural productivity, and even on environmental sustainability. Mycotoxin are secondary metabolites that are produced by fungi, which serve as their coping mechanism and for pathogenicity. Classes of mycotoxin include, we have the aflatoxin, we have the fumonisins that we'll be looking at today, we have the oxinivalenol, and so on and so on. The importance of this mycotoxin is because they cause severe disorders, both in animals and in human beings, and is really calling for intervention at this moment. So fumonisin that we are going to discuss today, or that we are discussing, was first isolated in 1988, and it contaminates both food and feeds. The regulatory, oh gosh, okay.
Sorry, let him again. I only reach the back up only.
Can you help us mute your? Can you help us to mute your device? If you are not speaking, we only allow our speakers to unmute and speak. Thank you.
Thank you very much, Chairman. The regulatory limit for fumonism in maize and its byproduct, as established by the European Union and Food and Drug Authority, is between the range of 200 to 4,000 milligrams per kg. The sources of fumonisins are series as of today that we know there are series which include the rice, the wheat, the barley, maize, etc. And also, with recent discovery, it was discovered that fumonisins are also a contaminant in white beans, black tea, and medicinal herbs. The producers of fumonisins are majorly our fusarium and fusariums and related other species like Alternaria also, which is found in grapes, raisins, wine, and coffee. It has also been reported that fumonisin is produced by Aspergillus niger. The impact of fumonisin, why are we treating fumonisin? Why is fumonisin of concern to us? Is that first, the International Agency for Research on Cancer classified fumonisin, especially the FB1, as group 2 carcinogen. That is, they have the probability of causing cancer in humans. Then the reduction in the competitive ability of our commodity in the export market, they reduce the appeal of the produce. Then also the consumption of fumonisin is linked to pulmonary edema syndrome of pigs, the esophageal cancer, neural tube defects, and it has been found that it also causes neurotoxicity, hepatotoxicity, and nephrotoxicity in mammalian and etc. It also impacts the sperm chromatin, which damage the motility and also reduce the DNA tail of rats. It was also found that consumption of this fumonisin affects the oocytes, which also impede the production of progesterone. So it also found that fumonisin causes liver damage in pigs, horses, rabbits, and primates, and it also causes brain damage in horses, kidney damage in rats, rabbits, and sheep. The consumption of fumonisin leads to low appetite or loss of appetite, which leads to a decrease in feed intake and conversion ratio. It also causes immune dysfunction, and aflatoxin and fumonisin also leads to changes in intestinal integrity, and it enhances intestinal pathogens. So now we can see that these fumonisins are not just a small joke. They are things that should actually catch our attention, because looking at it, causing this neuro, especially to the human consumption, causing this neural tube defect to human, and also affecting the oocytes, the progesterone secretion and all that, you could see that if care is not taken, fumonisin is a ticking bomb that likely one day, if care is not taken, is going to wipe off human generation, and not just human generation, even with our plants and animals, because we survive on consumption of both plants and animals, and we could see that the infection actually cut across both plants, animals, and even man is not left out in this impact. So the intervention strategy, what are the intervention strategies that have been put in place all this while? Because fumonisin is not just new. Like I earlier said, it was discovered in 1988. So what are the things that have been done before now in controlling or mitigating this fumonisin contamination? One, it's the development of resistant variety, breeding for resistant variety. Then the second one, use of fungicides. And we know that fungicide is already calling for attention due to development of pathogen resistance to these fungicides that have been used over and over again, its environmental impact, and the hazardous impact even on man itself while consuming. So we could see that we are trying to get rid of one problem and we find ourselves in another problem while we are using fungicides or chemicals in controlling it. So this is a defect in the control of fumonisin. Then we went on the back control, which we use antagonist organism, such as the bacillus and the streptomyces. These are actually viable, but its applicability on the field it's not really visible. Most of the studies about the back control of this pathogen, fusarium, is majorly in the greenhouse. So we are actually looking out for, okay, a sustainable control measure, a sustainable strategy that we can use to mitigate this pathogen or the mycotoxin that is being produced as fumonisin. So they also, with back control, the use of clay, absorbent, and use of essential oils such as ocimum. Cultural practices, manipulation of harvest time, in which early harvesting and all that has been put in place. Then also the post-harvest storage management to counteract our mycotoxin contamination by reducing the moisture content of our products, especially the maize or the cereals, to less than 50%, and manipulating the environmental condition of the storage. The contamination also is another strategy that has been used, which it includes the physical techniques such as dilution, that is mixing of different population, the one that is contaminated and the uncontaminated one, to actually bring down the concentration of the fumonisin in the population. Then the irradiation, which is actually adopted in the Europe with the use of the gamma radiation, the X-ray, and the electron beams to actually kill the mycotoxin or eliminate mycotoxin from the agricultural produce. Now, to the modern intervention strategies. Now that we are in the era of biotechnology, through identification of fum biosynthesis pathways, once it is being identified, then protocols have been put in place to ensure the degradation of the biosynthesis pathway. So the target is towards the biosynthesis pathway. And once we can actually target the biosynthesis pathway, alter it, then the production of this fumonisin will be degraded. Then an additional strategy, which includes the host-induced gene silencing. Gene silencing that is going forth for the gene of the fumonisin and actually finding a way to silence it and bringing in our own novel gene that we want into it. Then also the RNAi via the double-stranded RNA synthesis, which is also effective in combating these secondary metabolites. Then the most recent one, the omics, which is recently on ground, that is transcriptome profiling, genotyping profiling, in which the genes are being profiled to know the quantity or the upregulation or the downregulation of the genes to be able to actually modify our control towards it, to target the gene, to know if our the target of the control we are using or the strategy we are using actually has an impact on the gene, the genetic outcome, the genetic expression of the fusarium itself, the fusarium pathogen, the gene it's being expressed that controls this secretion of mycotoxin. Then also we have the CRISPR, which also goes along with the deletion of the FUMS biosynthetic gene and the modification of it in order to suit our own ideal. Then the metagenomic analysis, in which the microbial communities are being the microbial communities are being collected, and there's a screening that is being done. That is the soil. You know, our Fumonisins is a soil-borne organism. Our Fusarium, sorry, is a soil-borne organism. So the microbial organisms are being collected, and analysis is being carried out. Metagenomics analysis is being carried out to identify the genomes and to identify the contaminant areas so that it could be avoided or combat the appropriate combat option will be put in place. Then the application of hyperspectral and optical imaging, which is the most recent one, which uses capture spectral footprints of grains and nuts using deep learning to instantly classify and to sort out contaminated from non-contaminated products. So using this hyperspectral and optical imaging, it is easily, after harvesting our products, it is easy to be able to do the sorting out, and once the toxins, the one contaminated is being noted, it's being separated from the population. Then electronic gnosis and sensors, which uses pattern recognition algorithm to analyze volatile organic compounds emitted by modes, and it's used at the field level for detection of spoilage at the field level. So, and also the AI-driven transformation databases, which employ machine learning models like enzyme predictors to screen and discover specific enzymes capable of breaking down and detoxifying mycotoxin. So the function of this AI-driven transformation is actually to just read those enzymes that are capable to break down the fumonisin and detoxify the fumonisin. So once these enzymes are being found, they are now being put in, like loaded into the population to decrease the fumonisin contamination. Then the pre-harvest predictive modeling, which includes the integration of machine learning algorithms such as the random forest, neural network, and support vector machines with real-time weather, soil moisture, and all that, in order to detect the fungi contaminated zones and to be able to avoid them. So this is more or less like avoidance strategy to be so as to get rid of this fumonisin contamination. Then also we move on to the nanotechnology approach, which in our recent study we used selenium nanoparticles was biological synthesized using endophytic bacterium, and we found that selenium nanoparticles effectively suppress the growth of Fusarium graminearum. The essence of this strategy, the nanotechnology that we use is actually like it targets the organism itself. And it's once the organism is targeted and we are able to combat the organism, sure, the production of the fumonisin or the mycotoxin is being brought down, is being decreased. So the antifungal mechanism of this nanotechnology approach, or this selenium nanoparticles that was used, was discovered that it was through the disruption of cell wall, the induction of reactive oxygen species, that's the ROS, then the downregulation of the FGTRI genes, which is responsible for Dawn mycotoxin synthesis. So here is our result. There was a decrease in the production of Dawn by the Fusarium graminearum, and also the expression of gene. Using the QRT-PCR, we found that the expression of gene of the FGTR11 and FGTR14, FGTR15, and FGTR16 of that encompasses this fumonisin actually decreased as a result of our treatment with selenium nanoparticles. So the figure 1D that we are seeing there, it shows the mechanism that the ROS expression, the ROS expression, then also the toxisome. We also found out that the toxisome also actually decreased due to the use of these selenium nanoparticles. So the next experiment that our research group actually carried out, we also used zinc oxide. We synthesized zinc oxide nanoparticles using our endophytic bacterium. And using that endophytic bacterium, we got the resultant nanoparticles were characterized using the FTR, UVs, SRDT, EMS, EMEDS, and what have you. Then we found out that this zinc oxide nanoparticle was also able to exhibit antifungal efficacy. It impeded the fungal growth the colony number, the spore development, and also decreased the mycotoxin, that's deoxynivalenol, it actually decreased DON. Then the result of the confocal microscopy also showed that zinc oxide inhibited the penetration and the colonization of Fusarium gramineum in wheat plants. So this result actually also put together the mechanism. We studied the mechanism and found out that it's also through the ROS, the IRS that was released actually interacted with the cellular organelles and caused a damage to them. So thereby bringing down the population of our pathogen, that's Fusarium. So here, the figure 2, it shows the nanoparticles that was synthesized and the characterization that was done of the nanoparticles to be sure that our nanoparticle was successfully synthesized. And we used it and we found out that it actually also decreased the DON production from over 25 to less than 10 milligram per gram. Then also the major one is the destruction of the zinc oxide on the hyphae. We all know that the hyphae, the fungal hyphae, is used for colonization. So we could see that from this image, the 0 milligram, we could see the normal shape or the normal structure of our fusarium. But however, due to the application of 20 milligram per mil of zinc oxide nanoparticles, it caused a disruption. We could see twisting of the hyphae. From the SEM images, we could see the twisting, the wrinkling, and the breakage of the hyphae. So yeah, literally it means that the fungi, the fusarium, is unable to colonize successfully into the wheat plant. And the last slide, which the TEM slide at the lower part of the figure 4, you could see that the cell breakage. So hence, due to this cell breakage, there's exodus of the cellular materials. So all the cellular material, the plasma membrane and all that, actually excluded out of the cell membrane. So making the fungi useless. It could not actually perform its pathogenicity activity. So also, we now went also to measure the fluorescence due to the ROS that was seen, the fluorescence, this fluorescent exhibition. So we measured it and we saw that with the application of 20 milligram per mil of this zinc oxide, the ROS fluorescence was higher at 20 milligram compared to when there was no control. So without control, that means the fungi was still able to carry out its pathogenicity and produce mycotoxin or the mycotoxin, the DON. So, but with the application of these zinc oxide nanoparticles, we could see that it actually altered it and caused a destruction through the ROS. And then our recent research using bio-nanosensors. In this research, silver silicon nanosensors were synthesized, and figure 7 is showing the characterization, showing the successful synthesis of the silver silicon nanosensors. So after the silver silicon nanosensors were synthesized, we went on to use it as a bio-nanosensors. We applied it to in vitro into our plants, our bayberry seedling. That's 24 hours after inoculation. We applied it to see if these bio-nanosensors can actually detect the spectra or the stress spectra of the pathogen. So to our surprise, it was able to detect it even at 24 hours. It detected it, and we were able to pick the peak for it. So we used different treatments. The magnesium oxide bioprotectant was used in this case to protect the plant against this pathogen. And we could see that the spectrum 1454.46 is assigned to this pathogen, which is a stress signal. So as with the progression of the day, when we studied this spectrum, we saw that as the day progresses on the inoculum treatment, you could see that the spectra increases as the day increases. However, with the application of our magnesium oxide bioprotectant, as the day increases, the spectra for the stress or the pathogen was actually decreasing. So we found out that the magnesium oxide bioprotectant was able to protect our seedling, our bayberry seedling, from the pathogenicity of this particular fungi, which is Staphylococcus versicolor. So then we went ahead to note that the fingerprint of this fungus is actually at 1,454.46. So this research is actually important because on our for field experiment and all that, it's essential for us to detect the pathogen causing this mycotoxin or producing this mycotoxin on the field, and we should actually be proactive to combat the mycotoxin producers before it now translates or move into the storage. So once we're able to take care of it at the field level, then at the storage level, I think we will be safe. So this bionano is actually an important aspect of the modern strategy that should be employed in conjunction with artificial intelligence. It should be coupled with artificial intelligence to actually have a better and a futuristic combative strategy to be able to, and a successful one, which can be applied to other plants or seedlings. So now, this Pestalotiosis versicolor is also an endophyte, which I just discussed that we use the bio-nanosensor for, that causes that causes twig blight disease in bayberry. And in our recent studies, while we were trying to understand the virulence, we decided to carry out a Maditov analysis. And to our surprise, we were not expecting it. We found out that this fungi, Pestalotiosis versicolor, actually produced these fumonisins. It has not been recorded anywhere that Pestalotiosis versicolor actually produces fumonisin. So it now brings us back, take us about that. So now fumonisin is not just about contamination in series. Now it has gone wider to other crops, other fruits. So now it's more or less like urgent attention is needed. It's no more series that we are trying to avoid now. Even our fruits that we are consuming now has the probability of having this fumonisin in it. So this calls for urgent research for all researchers to go in to actually analyze the secondary metabolites of any endophytic fungi that they are working on to be sure that it doesn't produce fumonisin or any mycotoxin that could be of detrimental impact on man or on animal. So my conclusion, this fumonisin, it constitutes threat to global food safety and it demands a shift from our reactive measure to a proactive integrated management strategies, such as the back controls and the AI-driven monitoring. Effective mitigation requires immediate collaboration, not just the researchers, but a collaborative work with the researchers and the industry in order to implement this modern solution so that it will not just end up in our desk or in the website, but actually to implement it, our industry to actually implement. So we need a collaborative work for us to achieve this. Then conclusively, pending the time we get a lasting solution to this fumonisin, it is advised that there should be diversity in our dietary, that we should consume less of the series or the crops that are known to be susceptible to this mycotoxin, and go for the ones that are of lower risk assessment to these fumonisin. Yeah, these are my selected references. Thank you.
Thank you so very much, our distinguished presenter, Dr. Ayade. We so much appreciate you for that insightful, for the knowledge sharing about fumonisin from silent threat to smart prevention. You made us to know that mycotoxin are, these particular fumonisins are classified as group 2B carcinogens to human, and taking bomb require urgent intervention. And from your presentation, which highlights a shift from conventional controls such as fungicides toward sustainable integrated strategies, resistant crops, biocontrol, moisture and cultural management, and appropriate physical salting separation. This takes us also to the next generation of control, which is technology-driven, RNA, omics, metagenomics, hyperspectral imaging, electronics, noise, sensor, AI-powered database, and predictive modeling for early detection and prevention. And this is an exciting frontier in nanotechnology, which includes zinc oxide nanoparticles and biological endophilic approaches with promising antifungal protection. Yeah, this message is very clear. Fumonisin control is moving from simple detecting contamination to predicting it, preventing it, and intervening early with biology, molecular science, sensor, AI, and nanotechnology. We so much appreciate you for everything that you have just presented to us within a very short period of time and broaden our knowledge about fumonisin, bringing us back to what we actually need to focus on. We thank you for dedicating your time for this presentation. On this note, I would like to hand over to, if there is anyone that wants to ask question, please kindly use the chat box. I said people should send text messages. Okay, now we have some questions. Number one, somebody sent a question. He said, "Thank you, Dr. Ayuade. I feel like taking the lecture over again." It says, is pestalotosis vascular found or has it been reported in Nigerian food? The fingerprint identified in the bionano sensor experiment, does it apply to a specific species of fumonisin or can it detect other fumonisin? This is one of the questions. This is number one questions that I've been able to gather. If anybody has other questions, you can just raise up your hands and we will call you. Very brief question. Thank you. Over to you, ma'am.
Thank you very much, ma'am. And in response to this question, actually, Pestalozzosis versicolor is discovered in China, and its effect is more in the Chinese region. And the reason why this pathogen is of importance is because China is one of the highest growing community for bayberry. And bayberry itself is used in production of wine, jam, and also it's consumed as just as the fruit as it is. So the population of Chinese, it's really high, it's really large. So having that pathogen affecting their bayberry, it's actually of concern. But however, I've not found pestalozzosis, or I've not read about, not found, I'm yet to read about pestalozzosis versicolor in Nigeria. I'm yet to read about it. But my current knowledge is found in the European countries and in the Chinese, but in China, but not in Nigeria yet. Then the fingerprint identified for the bionano-sensors. This fingerprint is actually for identifying the pathogen, not for the fumonisin. Is to identify the pathogen itself, which is the pestalozzosis. So once the pathogen is identified, then you can actually eradicate the pathogen. So the work towards is actually for the bionano-sensors, it's actually a fingerprint in identifying the pathogen itself. So once the pathogen is eradicated, then we know that the mycotoxin that is going to be secreted is already gotten rid of. So it was actually channeled to identifying the pathogen. And yeah, thank you.
Thank you very much. I hope she has been able to answer the question according to the way you wanted her to answer it. To me, I believe she has, but I just want to know. We appreciate you for that. And again, another question, what are the major realistic management practices of fumonisin and fumonisin by local Nigerian farmers? If there is anyone, because we focus more on aflatoxin. But now somebody is asking if there is any realistic management practices of fumonisin and fumonisin by local Nigerian farmers.
Now the attention is going towards nanotechnology. And I would say nanotechnology is actually the realistic one, because herbicides and chemicals are no more safe for us to use. So nanotechnology is now the heat of the day to be used in controlling it, and it has been used on experimental sites, and it has been found to be very, very good in combating pathogen, especially this fusarium and the fumonisin. So I will recommend nanotechnology. Then also the back control. We know Nigerians are into back control, but however, this back control on the field, I've not the impact on the field. due to the environmental condition, the differences in the environment and all that, it might not be so realistic to apply it on the field because of the varying environmental condition. So birth control might not be actually a deal of the day. But nanotechnology, they are always like, it's, how will I explain this like nanoparticles, once it's synthesized, it comes either like it's in a powdery form and you can actually like dissolve it into your soluble solvent, then you can apply it as spray or what have you, as foliar spray on your crops. So nanoparticles, application of nanoparticles is visible in Nigeria market or in Nigeria farming of today. Thank you.
Thank you very much. We appreciate you. The first person that first asked question sent his thank you. And okay, and the same person still asks another question. He say, how soon can the nanoscience solution get into the market for wide application? What are the potential setbacks? I think for now, we should limit our questions. This is going to be the last question that we take. How soon can the nanoscience solution get into the market for wide application, and what are the potential setbacks? Thank you.
Nanoparticles application, it all depends on our acceptability. Because whatever new idea or new science that we are bringing in, it all depends on the acceptability by our farmers. So that is the determinant. The acceptability by our farmers is the determinant. Once they're able to accept it, or once they are aware of it and it's a welcome idea by them, then it's good to go into our Nigerian markets.
Okay. Thank you very much. We appreciate you so well, Dr. Ayode. We really appreciate you. Thank you for those very important responses. And I believe today's presentation and discussion has reminded us of one fundamental reality, the challenge of fumonisins, which require us to move beyond detection and documentation towards prevention, innovation, and action. We know that the knowledge exists, but the technologies are advancing. What is increasingly important is how we translate that knowledge into practical intervention that protect consumers, support farmers, and strengthen food safety. Thank you so much. We want to thank you for everything. On this note, once again, I would like to please, I would like to please say that we're sorry for starting so late. It is not in our culture to do something like this, but you know, at times you cannot be in control of internet. I guess that was what happened. We kindly, I apologize on behalf of the executive members of Mycotoxicology Society of Nigeria. So on this note, it is now my pleasure to invite Dr. Sahib Shitu. He is the Assistant Secretary General of Mycotoxicology Society of Nigeria to deliver the vote of thanks and closing remark. Thank you. Dr. Shitu, please, you have the floor.
Well, okay, thank you.
Hello, Dr. Shitu.
Moderator, can I go on?
Yeah, you can
Dr.
go on.
Shitu.
Okay, ma. Good evening, everyone. Let me first of all appreciate Almighty God for giving us this wonderful opportunity to have this webinar. And at the same time, I most sincerely appreciate our guest speaker, who out of no time, she created time to be with us despite the one or two challenges we had before the commencement of the lecture or of the webinar. We are really grateful. We really thank you for your time and for this vast knowledge you shared to us. I must confess that we have learned a lot. And I also, on the same line, thank our amiable president in person of Professor Charles for his wonderful encouragement and initiatives. And at the same time, also, I want to thank the coordinator for this webinar in person of Mrs. Talatu Ethan. May God continue to preserve us all, and may He continue to protect us and guide us aright. And for our participants, those that joined the meeting, we really appreciate you. And we hope for the our guest speaker, when next we sought for your time, you graciously accept our invitation and be with us. So it is my pleasure at this juncture to thank Almighty and all that participated and contributed in one way or the other. Thank you very much. Thank you.
Thank you so very much, Dr. Shitu. We appreciate you. On these notes, we want to thank the Almighty God who has made this webinar, the 6th International Webinar of Mycotoxicology Society of Nigeria, who has allowed it to come to pass. We appreciate him. We appreciate everyone that has come. We thank our speakers and everyone, most especially the president of this association, and also the director of research, that's Mrs. Talatu Etan. We want to thank everyone. Thank you very much. On this note, the 6th International Webinar of Mycotoxicology Society of Nigeria is hereby brought to a close. Thank you all, and God bless you.
Thank you, ma. Thank you all. Thank you for finding time. Thank you very much. We are deeply sorry for the delay. God bless you all.
Thank you
God
very
bless
much.
you all. God bless you all.
Amen.
Sama.
Amen.
Everyone, we are grateful. We are deeply grateful. And we are deeply sorry.
No problem.
Thank
Bye,
you,
sir.
everyone.
Bye,
God
ma.
bless you. Have a lovely day.
President, thank you for
sister
sourcing
Thank you,
for
our super mama.
facilitator for us. Thank you, sir.
Professor Jeff,
The
thank you,
president,
ma.
all the
We
prof.
appreciate Professor Johnny.
Prof,
Everyone, all the
thank you.
names. Thank you.
Our able moderator.
Dr.
Everyone, God
Shitu,
bless you
thank
all.
you. Thank you,
Bye,
everyone.
Dr. Shitu, everyone. All the
Thank you, ma.
participants, we are grateful. President, thank you for always getting facilitator for us. We are grateful, sir.
It's God, ma. We appreciate your
Thank you,
directorate,
ma.
ma. Many more
Thank you,
blessings,
sir.
ma.
Thank you, sir. God
Thank
bless
you,
you
ma.
all, sir. God bless you all.
Dr.
Bye.
Apia, I cite you.
Yes, so
And our
thank you,
PRO, what happened
President.
today? Oh,
Oh,
PRO,
no.
all is well. Bye.
Dr. Dalati, I cited you. Charo,
Everybody,
I cite
Sir,
all of you. Well done, no.
sir, I was not able to. I raised my hand up, but no question, nothing, nobody. I said, oh, it was a wonderful lecture, sir.
Okay, no problem. We are picking you on board very soon.
Don't worry, bro.
Where you
It's
will do all
okay.
the talking.
I asked you a question the other time, sir, but no response.
Oh, sorry, I've been moving, traveling. Even as I'm talking to you, I'm on another journey. So I will check and get back. Sorry, sir.
Okay. It's okay.
Yes, sir. Bear with me, sir. Sorry, sir.
Professor Chance, how are you?
Good evening, Prof. How are you, sir?
How weekend?
But Dadani, sir. Everything is fine, sir.
Oh,
Good
thank
day,
God.
sir.
God be the glory. Chief Danladi.
Yes, sir. Yes, sir. Yes, sir. Yes, sir. Yes, sir.
Ẹ ṣé.